Hey guys, welcome back to another episode of Beyond the Yellow Tape. My name is Julian. I'm glad to have you guys here with us again. And today we have our special guest again, Nidhi. Nidhi, tell the people what's up.
Hi, everyone.
All right. So I know I usually talk to you guys about the science behind everything, but I just want to briefly explain to you guys how the science and the courts and everything ties in. So uh, let's start from the crime scene, which you have your first unit of the triangle, we'll call it, which would be law enforcement, where you have your police agencies, investigators, detectives, officers at the crime scene, picking up any types of evidence, getting all the information necessary, getting the scope of the crime and whatever happened. And they're taking that evidence and pretty much that whole scope of the crime down to the next part of the triangle, which is the scientific analysis, where you have your forensic labs, your analysts just like me, and your research lab, which is where Nidhi's at, where they either analyze or in terms of a research lab, they introduce new methods or new methodologies of, doing work. So, they might discover a new way to find out if this is blood, a faster way. So they're always working on things like that. And then your last part would be the courtrooms, the legal proceedings, the court, judges, juries, where we test our findings and prove our findings right, pretty much. So I would say that is that would be the big trifecta right there for how everything ties in together. And speaking of crime scenes, I know last time I kind of want to show you guys how Nidhi, who knows a lot about DNA and DNA profiles, so how someone in my position might tie into someone in her position. So that would work with, say we have an officer at a crime scene and he sees a reddish brown stain, he doesn't know if it's blood. So he would bring that back to me in the lab as an analyst. But before he even does that, he would do something what is called a presumptive test, or he may do something what is called a presumptive test, in which he may test that reddish brownish stain to see if it's blood. And these presumptive tests are something pretty simple. It might be like a color stain or color change, just something really quick to make sure that it's blood. And then he may take it back to me in the lab, and I would test that to make sure it's blood with a confirmatory test, where we wouldn't have to worry about maybe something like false positives or false negatives as much as you would with the presumptive test, because the presumptive would be less sensitive. The presumptive test can be fooled by something like maybe animal blood or tomato paste and some horseradish and some bleach. All these kind of things can disrupt the presumptive test, but not the confirmatory test that we may do at the lab. So after someone like me does a confirmatory test in the lab, I would, and prove that it's blood, I would send that sample to Nidhi, our DNA analyst, for example. And then with that, she will make a DNA profile, and I'll let her take from here.
Thank you, Julian. So yes, absolutely right. So when the sample, which is after being collected from the crime scene, biological material comes to the lab or with the DNA analyst, there are mainly 5 steps that the DNA analyst has to perform before for getting DNA profiles. So even before I get into the 5 steps of the forensic DNA process, let me just explain a little bit what a DNA profile is. So DNA profiling is a forensic technique which basically helps us to identify individuals like you, me, or someone else based on the differences and variations they have in their DNA sequence. So this variations kind of makes every individual unique, except identical twins. So they have the same DNA profile.
Okay, that makes sense.
So that's basically DNA profiling. So it's not that easy to develop a DNA profile. There are, like I mentioned, there are 5 steps that a DNA analyst would have to go through. The first being DNA extraction. The second is DNA quantification. How much DNA you just extracted, you would wish to know. The third step is DNA amplification, which is this, you can make multiple copies of DNA that you are working with or looking for. Then it is separation of these copies that you just made. And the last step is the analysis of the profiles, the DNA profiles that you got through the entire, after you perform the entire process. And this takes a lot of time. Like each step in the lab has its processes, and it takes a couple of hours to do extraction, a couple of hours to do amplification. So all in all, it takes a lot of time. It's not quick and that fast.
So but it's not how it is on TV where they put something into like a computer and get the face of a beautiful man all of a sudden.
Oh, no, it's absolutely not easy. And it gets more and more complicated when you have DNA profiles from multiple people, like when you get a sample from the crime scene, which is a contribution of a lot of people. Then imagine you have a profile which has multiple individuals being analyzed together. It becomes very difficult for me or any DNA analyst to separate out, oh, this is the person it is, this is the person.
Right. Well, I know that in my little amount of DNA knowledge, that one of the ways is for one of the ways of extraction is meant for that, pretty much, right? To separate stuff out.
Yes, so that is called a differential extraction.
So that would be used more in like a sexual assault rape cases, right?
Yeah, exactly. Absolutely.
Okay.
So you don't, so the point of doing differential extraction in sexual assault case works is the same thing, that you don't profile a male and a female, a perpetrator and a victim together and have a DNA profile of both of the samples together. So you need to separate it out. Yes. Yeah.
That's good. That's cool. That's crazy.
Yeah.
So is there any other types of extractions, or is that the main one?
No. So there are basically three types of extraction. One is organic extraction. The second is Kilex extraction. And the third is FTA, or solid phase extraction. So the differential extraction that we talk about is a modification of the organic extraction part of the, yeah.
Okay.
Yeah.
Damn. And how long is the extraction phase? How long does that last out of the whole 2-day period? For how long would that take you?
So I do something called Qiagen extraction in the lab that I'm currently trained on. It takes about an hour or 2 hours for me to do the extraction, which is not that bad, absolutely. And then after, so extraction is, like I said, it's not the only step, right? After you get your pure DNA out. So I'll just, before I get into the other parts of the process, just let me get into a little bit about what extraction is. Because I think it would be nice for the audience to know what is extraction.
True.
Yeah. So what happens is, say, for example, you get a biological material, which is either in form of blood, semen, or saliva, or skin cells. You need to break the cell apart and the nucleus in the cell to get the DNA. So the DNA is in the nucleus. So to free the DNA from the nucleus of the cell, we have to perform the three different, one of the extraction process that I mentioned, which depends on the type of the body fluid. We use whichever extraction method is suitable for the biological fluid that we are working with. So our aim is to get the DNA free from all this, the proteins in the cell, free from the nucleus, and pristine DNA in your in the tube that you just performed your extraction. So that in the downstream analysis, when you do quantification, where you need to know how much DNA it is, or when you do DNA amplification, where you make multiple copies of the DNA that you just extracted, you want the DNA to be free from anything which causes an inhibition in the process.
So you're pretty much just cleaning it out. So when you, so for the next phase is pretty much.
You do get rid of the cellular debris and the unnecessary proteins. So so you want free, clear DNA. In through your extraction process.
Okay, that seems like a lot. So
It is a lot.
What needs to happen is next week, we got to have you back on so you can give us the deep dive into quantification, amplification, separation, and the overall profile. Does that sound like a plan?
Yes, definitely.
Right, that sounds like a plan. And I'm happy, I am pleased. I learned something new. Boom. So it's officially a good day. All right, peoples, you know the vibe. If you like what you saw, like what you've seen, like and subscribe. My name is Julian. If you need to hit me at J, the number 4, ENSICS 2020@gmail.com. And Nidhi is going to tell you how to contact her.
Yes, thank you, Julian, once again. And thank you, everyone, for listening us talk about the criminal justice system and a little bit into forensic DNA extraction. And if you have any questions, concerns, or suggestions, you can contact me at sheth.forensics@gmail.com. You can also find our emails in the description below. So please like, share, and subscribe, and stay tuned. And we'll see you in 2 weeks. Bye.
Amen. Oh, and follow the Instagram. It's at Beyond the Yellow Tape.
Yes. Follow the Instagram page.
Follow IG. Y'all get to know when we're gonna, drop and do stuff, and a bunch of stuff is in the woodwork. So stay tuned, guys.
Yeah, stay tuned, guys. A lot of interesting things coming up.
All right, guys. Peace.
Bye.